How do monoclonal antibodies target antigens for lymphoma? Imagine a tiny missile designed specifically to find and attack cancer cells. That's essentially what monoclonal antibodies do in lymphoma treatment. These are lab-made molecules engineered to recognize and bind to specific proteins on the surface of lymphoma cells. In non-Hodgkin lymphoma, these proteins are called antigens, often part of a group known as cluster of differentiation proteins. Common targets include proteins like CD20, CD30, CD19, and CD52, which are found on the surface of certain lymphoma cells. The process begins with testing the patient's lymphoma cells to identify which of these antigens are present. Once the target is known, doctors select a monoclonal antibody that specifically binds to that antigen. For example, rituximab is a widely used monoclonal antibody that targets CD20, a protein found on B-cell lymphomas. When administered, rituximab travels through the bloodstream and binds tightly to the CD20 proteins on the surface of malignant B cells. This binding acts like a flag, signaling the immune system to recognize and attack these cancer cells. Monoclonal antibodies can kill lymphoma cells through several mechanisms. One way is antibody-dependent cellular cytotoxicity, where immune cells such as natural killer cells recognize the antibody-coated cancer cells and destroy them. Another method is complement-dependent cytotoxicity, where the binding of the antibody activates a group of proteins called the complement system, leading to the formation of pores in the cancer cell membrane and causing cell death. Some monoclonal antibodies can also directly induce apoptosis, which is programmed cell death, without needing immune system help. In addition, some monoclonal antibodies are conjugated to chemotherapy drugs, toxins, or radioactive particles. These conjugates deliver potent agents directly to the lymphoma cells, sparing most normal cells. For example, brentuximab vedotin targets CD30 and delivers chemotherapy directly to the cancer, while ibritumumab tiaxetin delivers radiation to the tumor. There are also newer types called bispecific antibodies, or T-cell engagers, that can bind both to a lymphoma antigen and to T cells, bringing immune cells into close contact with the cancer to enhance destruction. Typically, monoclonal antibody treatments are given through an intravenous infusion over several hours in a clinical setting. They are often combined with chemotherapy to improve the overall effectiveness of treatment. The choice of antibody depends on the specific antigen profile of the lymphoma, making the treatment personalized for each patient. In summary, monoclonal antibodies target lymphoma by specifically binding to proteins on the surface of cancer cells. This targeting flags the cells for immune attack, directly triggers cell death, or delivers toxic agents to kill the cancer. This precise approach helps minimize damage to healthy cells and has become a key part of standard lymphoma therapy. As research advances, scientists continue to develop new antibodies and improve existing ones to overcome resistance and enhance treatment outcomes. Always consult healthcare professionals for personalized advice, as individual responses to therapy can vary.